Morphological and anatomical study of supply specimen, that is the Cycas stem and Cycas leaflet, and these things have to be morphologically analyzed and as well as anatomically, so that we can get some of the internal structures of as well as the morphological characteristics of this species. So here the aim of the experiment is to study the morphological characteristics as well as anatomical behavior of the specimens. As we know, the cycas is distributed and different species of cycas are found in different geographical regions of the area. So each species is not possible to collect from one area, and on the basis of different area, different species are found. And we can have we can buy these species as a bottle specimen for our practical purposes, and different species locally available cycas species are there in our local area also. For the morphological as well as anatomical study in the laboratory, we need some materials for study in the laboratory, such as needle, blade, safranin, methyl green, microscope, slide, cover slip, pen, paper, plant specimen, etc. So these are the things we need for the analysis for the study. And for the morphological study, we have to observe, we have to collect the plants from the field, and we have to observe the habitat of the plant where the plants have been growing, and note down the other characteristic features of the field, at what positions they are in the field. And we have also to observe the reproductive structures which are found there in the field in the plant. For anatomical study, we have to make sections, that is transverse sections of the each specimen, that is stem as well as the leaflet, and take separately each of them to is transferred and processed through double staining process and observed under compound microscope, and noted their anatomical characteristics features. For the double staining process, we have to use safranin and make the sections and place the sections in the safranin first for 1 minute, 1 to 3 minutes, and then we have to transfer the sections to 70% ethanol for 1 minute, and after that the sections after that the ethanol will be soaked with the help of blotting paper, and again 70% ethanol is added for 1 to 2 minutes, and after that the section is transferred to methyl green solutions and keep for 1 minute. And immediately after the staining with methyl green solutions, we have to convert the sections to 95% ethanol and for 1 minute, and again up to 95% ethanol for 1 to 2 minutes, and blot out the ethanols. So we have to remember one thing, that we don't have to leave the sections for too long in ethanol, as it may remove too much of the stain. The section should appear translucent to blue. So if it appears red, then the section stain has not been effectively removed. At the end of the method, we have to gently transfer the section to a microscopic slide and with filter paper blot out excess ethanol.